Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to the TV learning series as you are used to study using TV programs. I'm teacher Hurbert. I will be taking you through the biology lesson of today. Student, the biology lesson of today is a biology practical lesson. And I want to remind you to take your exercise books, notebooks, and you try to write somewhere the important notes of what we're going to do today. Students, that is the lesson of today. So, in biology practical lesson of today, we are going to test for the nutrients. I think last time we discussed about testing vitamin C, we talked about testing fats, and now we're going to test about the reducing sugars and non-reducing sugars. Examples, who can give me an example of a reducing sugar? An example of reducing sugar that we're going to be using today is glucose. Then another one, I'm going to test non-reducing sugars. If I say non-reducing sugars, you're going to have sucrose. If I say sucrose, we can have examples of sucrose. We can have orange, we can have carrots, we can have sugar canes. These are examples of reducing sugars. And you know, when you talk about reducing sugars, glucose is an example of monosaccharide. It is made up of one molecule of glucose. And then non-reducing sugars, these are disaccharides. If I say disaccharides, examples of disaccharides are sucrose. We can say it is made up of two molecules of sugars. So students, let me just welcome you to the practical lesson. The practical lesson I'm going to be having in front of me, I have a decorated table which has the solution, the reagents. So the solution I have, it is a prepared glucose. I used biscuits. You can use biscuits. You can use sugar. And then we start preparing the reducing sugars. I have sample 2, which is composed of non-reducing sugars, and I used sucrose as an example of non-reducing sugars. I have sample C. As you can see, this is sample C. I'm going to be using a distilled water. If I say a distilled water, it is a water which does not contain any impurities. So the distilled water is going to be acting as a Contra experiment. If I say a Contra experiment, it is an experiment that controls negative, not test positive. So let us start the experiment. I'll be using the reagents, chemical reagents. Let me just write the reagents that we are going to use. Reagents, or we can call it the chemicals that are going to be used. I'm going to use one, which is supposed to be the Bendix reagent, or you call it to be a solution. I'm going to use number 2. I will use hydrochloric acid. That is the symbol of hydrochloric acid. Hydrochloric acid. I will also use 3. I will also use sodium hydroxide. Sodium hydroxide. This is sodium hydroxide. You know the reason why I have to use hydrochloric acid and sodium hydroxide. I will explain it in the process. Let's start our experiment. You get a test tube. After getting a test tube, you put it into a test tube holder. After putting it into a test tube holder, you get a sample of the solution 1, and solution 1 is glucose. That glucose should be well prepared in the All right, let me put the gloves to protect myself. So from there, I'm going to get a sample. The sample I'm going to be using, I will use the equal amount of the solution. You get like 2 centimeters of the solution. The solution, you put like 2 drops. Then from there, you add the bendic solution. This is the bendic solution. After adding the bendic solution now, you get a dropper. After getting a dropper, you get the same amount, the same drops that you used, your dropwise. Dropwise, and you add the drops of the drops of the bendic solution, and you can see the color of the bendic solution is blue in color. So we are going to see the observation now. You shake it slowly, and you put it into a water bath that contains boiling water. We are going to start observing and see what are the color that should be changing, and that's what would be the indicator that show that test positively that our sample contains reducing sugars. Let us try to observe and see. And we should just be observing using the color. And once you are making the observation, you do the observation, you do the conclusion or the deduction. So let us try to observe. I think as you see, the color which was blue in color is trying to change from blue to green. So it is changing from blue to green, then from green to yellow, and finally to moderate orange or to orange precipitate. You can have orange precipitate, or you get moderate orange precipitate, or you can also have what you call brick red. So let me just try to, as you are seeing, it is changing to green. We are observing it is changing from blue to green. So let us try to note something on a chalkboard, and then we write it. Meanwhile, when we need to wait and see if changing from green to yellow and finally to orange or to moderate orange. So I'm going to observe the changes that has happened. Of course, everybody can just make an observation and make a conclusion. We see what color do we observe from here. It has changed from blue to green, then from green to yellow, then finally to orange. Or you can also sometimes you can also observe the brick red. Thank you. The second practical, we are through with the reducing sugars. Let's talk about non-reducing sugars. The non-reducing sugars, I said today we are going to use sucrose as a sample. We are going to be testing the presence of sucrose in our sample. I have a prepared sample, extract from, that one is made from the industry, But you can also use the carrots, you can also use the oranges, you can also use the sugarcane, and we extract what you call a sample that contains sucrose. So from there, let me start my practical. Before, let us say, why do you have, how can you just go about it? When you are testing the non-reducing sugars, we use also the Benedict solution. But adding the Benedict solution, the sucrose is enabled to reduce the copper ions. So the color of the Benedict solution and the sucrose persists, it doesn't change. How can we now break the sucrose into a monosaccharide? You get the sucrose and you add procedures. We have a duster. That is, we are now on number 2, which is reducing sugars. Let's talk about the procedures. Procedure number 1, you get a sucrose sample. Then from there, number 2, what do? You from the sucrose, you add HCl, hydrochloric acid. In bracket, you can say hydrochloric acid. And I can say hydrochloric. Why do you have to add hydrochloric acid? I told you sucrose is a disaccharide. So once you add the Benedict solution, the color of the Benedict solution, it persists. Why? Because we know the sucrose is enabled to reduce the copper ions that are present. So what we normally do is to add hydrochloric acid to hydrize the bonds. After hydrizing the bonds, it breaks the bonds from a disaccharide to a monosaccharide. You get the point? 3, you boil the sample. Then 4, after boiling, you cool the solution. After cooling now, we go to number 5. After cooling, what do? You add drops. You can say drops of sodium hydroxide. Now, number 6, what do? You observe color changing. Let's try to do the practical now. I said, now there is something also we have to talk about. We also have to remind ourselves, why do I have to add sodium hydroxide? I add sodium hydroxide in the solution purposely to neutralize the acid present in the solution. To neutralize the acid present in the solution. Let's now do the practical now. Quickly, let us do our practical. This is a sample which is containing reducing sugar. I also use a sample of distilled water to act as a Contra experiment. I will get 2 test tubes. Test tube 1, if I have a marker, I could be labeling them. Test tube 1 will be containing a solution containing sucrose. Test tube 2, I will put there distilled water, which is going to be acting as a Contra experiment. So let us do our experiment. I get the sample. From there, then after adding there, then I add hydrochloric acid. Let me just first do one thing. I add hydrochloric acid. This is sodium hydroxide. This is hydrochloric acid. Then I get hydrochloric acid. I add some drops of hydrochloric acid. The drops of hydrochloric acid Then after putting there, I need this. Then I have put it into a water bath. Into a water bath. From there, here, we are in a kitchen. We are trying to see, not in a kitchen, we are in a laboratory. We are preparing the sucrose to test if the solution contains the non-reducing sugars, as an example of sucrose. When you heat, automatically you have to heat. After heating, you cool the solution. After cooling the solution, you add the bendic solution. And then finally, you heat it again. So observe the process. I'm now in a process of heating sucrose. I have added hydrochloric acid. Observe properly, and then we see the color changing. You cool it, put it into a warm water. After you put it into a warm water, the next is to add the bendic solution. You cool, you take like 3 to 4 minutes of cooling, and then after you remove the sample, you add a bendic solution. I think it is cool. You add now the bendic solution. Add bendic solution. After adding a bendic solution, you add sodium hydroxide. You get sodium hydroxide. You add some drops of sodium hydroxide to neutralize the acid present. And then you put it into a water bath again. You heat. In the process of heating, we try to observe what is going to take place. Automatically, it has to change from one color to another color, and finally, we're also going to have orange precipitate. Good. Student, I think when we are making the observation, we try to see now. Even you can see there is orange precipitate that is going to be formed, meaning that there is presence of reducing sugars. There is presence of non-reducing sugars. You can see the color is changing from the blue color to orange precipitate, which indicates there is presence of non-reducing sugars. Non-reducing sugars. I think finally we can see we have the color is changing to orange, and which indicates there is presence of non-reducing sugars. I said, why do you have to add hydrochloric acid, student? I said we add hydrochloric acid to hydrize the bonds, because I told you the sucrose is enabled to reduce copper ions. So we have to add hydrochloric acid in order to hydrize the bonds. Then from there, we add sodium hydroxide to neutralize the acid. I think we have got the final answer, which is supposed to be, so meaning that our sample contains non-reducing sugars. Meaning it has changed from blue to yellow, then from orange to yellow, then finally to orange, which indicates the presence of non-reducing sugars. So we can say color changing. You can say the sample, I can say sample 2 contains non-reducing sugar, non-reducing sugar, because the solution, the sample, changed from blue to orange, and then to yellow, then finally to orange, which indicates or which confirms which confirms that there is presence of non-reducing sugar. Non-reducing sugar. Or there's presence, we can say, there's presence of sucrose. So I think that is the sample we had to do today. Quickly, I'm telling you that we can say that our sample, we had sample 1, which was reducing sugar. We had sample 2, which was non-reducing sugar. And I said we can use this one to test the presence of non-reducing sugars and non-reducing sugars. Students, I think I can give you an assignment or a homework. The lesson, I think, is very well understood and very clear. So I think I can give you an assignment and a homework of which we can do together. So this is the assignment. You can say carry out the following tests. Carry out the following tests on extract A using the reagents provided, and record your observations. Record your observations in a table shown below. So let us have the table. Let's have the table. Let's have the table, and we discuss briefly what we're going to be doing in that table. I can say this is the experiment. These are observations. And here, the third column, we have deductions or stroke conclusion. One to 2 centimeters cubed of extract A in a test tube. Comma, add 3 drops of iodine solution. I think we did the test of starch at the beginning. Question 2, to 2 centimeters cubed of extract A in a test tube. In test tube, add hydrochloric acid and add 2 centimeters cubed of bendict Benedict's reagent, cool the solution, and add sodium hydroxide. And then three can say from the table there, that is Roman 3. This is Roman 3. You can talk about no. Let us screen. Let us screen very quickly. You can say this is experiment, observations, then conclusion. Then I can say, you say add 2 centimeter tubed in the test tube, comma, of extract A, add Benedict solution, and then you make the observation and the conclusion. The last one, you can also talk about add 2 centimeters of extract A in a test tube, extract A in a test tube, and add burette solution. Burette is burette solution. Then make the observation and the conclusion. So you can see they are almost, as you can try to observe properly, there are 3 experiments. This is Roman 1. You add this one, and you make the observation and the deduction. This is question 2. You do observation and conclusion. So experiment 3, this is 3, Roman 3. You also do the same up to Roman 4. So I've mentioned about the 5 steps of how to test the non-reducing sugars. I said number 1 is, as I mentioned. Then number 6 we're going to do is observing observation of color changing. So that is step 6. When you add sodium hydroxide, the step is this. We are going to see the color will change that if there is presence of non-reducing sugar, that is, say, i.e. sucrose, the color changed from blue to yellow, then finally to orange. This indicates or confirms the presence of sucrose or non-reducing sugars in the food sample. And when it is absent, when you use the Contra Experiment, like water, the blue color of Bendix solution persists, does not change. So it tests negatively. So students, I think the lesson was okay. Thank you for being with me in my lesson. Thank you for active listening and noting something. I also thank you and I remind you to stay home and to wash your hands with soap and clean water.